Good morning. Let's discuss how your skeletal muscles work. Okay, so we're going to first look at how it's put together anatomically, and then we'll discuss how the filaments move to create muscle contraction, meaning to cause the muscle to shorten. So we can begin with, again, the structure of a typical muscle. So a whole muscle that you see, the whole muscle is called the whole muscle here. Like, for example, your biceps brachial muscle is a whole muscle. That muscle is compartmentalized into bundles called fascicles. And then these fascicles contain numerous muscle cells. And these cells contain many smaller things yet called myofibrils. And then myofibrils contain in them smaller components yet that we call myofilaments. And it is at the level of the myofilaments where movement occurs. So let's look at myofilaments. So your myofilaments come in two types. You have your thin myofilaments, which contain three different proteins. You have proteins on them called actin. So your actin proteins that make up your thin myofilaments. You also have proteins on them. So these these actin molecules have on them a binding site for myosin. So these are your myosin binding sites there. And then covering these myosin binding sites is another protein here that we call tropomyosin. This is tropomyosin. And then there's a third protein hanging out here. This protein is called troponin. So the role of tropomyosin is to cover the binding sites. Cover the binding sites on actin. And the role of troponin is to remove tropomyosin. In other words, its job is to unblock actin, okay? And it does so by another role it does is it binds calcium. So the three, this is what troponin does. It binds calcium in order to remove tropomyosin in order to unblock actin, okay? And then your thick myofilament contains its own protein, a singular protein called myosin. So thick myofilaments, bad marker here, I'm in class again. Thick myofilaments contain a singular protein in bundles. And that singular protein is called myosin. It has these things on it, these heads, okay? So this is myosin there. And this is the head of myosin. The head of myosin. And that head does a few things. So the head has a part called ATPase. That's an enzyme whose job it is to convert ATP to ADP plus PI. That's what that enzyme does, ATPase. which is an enzyme in the head of myosin. It is also a part of the head that binds to actin. So there are two jobs that myosin takes care of. It, one, converts ATP to ADP and PN. It's called the ATPase part. And secondly, it binds to actin. It grabs it. So let's look at how these thin and thick myofilaments are arranged inside of your myofilaments, inside of your myofibers. Let's turn this board over. See if we can do it today. I'll leave my hard time getting here. Very good. Okay. So let's arrange your thick and thin myofilaments in what we call sarcomeres. So sarcomeres is just the units of arrangements of your myofilaments. Excuse me. Let's stop up here. So let's begin with your actin. So actin here, actin. And so over here, actin. Here again, actin. So you always have your thin myofilaments overlapping the thick ones. So here in the middle, we would have your thick myofilaments here. And that would have, its extensions. Of the heads of myosin like this. So here's myosin. Myosin with the heads like that hanging out next to actin. And these myofilaments, the thin ones, are linked together by a protein called the Z disc. This is the Z disc, which anchors the thin myofilaments together. This is your thick myofilament. This is thin. Okay. And this arrangement of thin over thick is called a sarcomere. And these will be attached together this way as well as this way along the length of the entire myofilament. You can review your anatomy notes here to see how this is arranged if this is not clear enough for you. But I wanted to show you this so we can get to the next step to explain how it is that these two proteins interact to create movements of the muscle. Okay, so these next steps that we will discuss is what we call your sliding filament mechanism. All right, so let us erase and start again. So we're going to discuss how the thin and thick myofilament interact with one another. Okay. So begin with, here this is called your sliding filament mechanism. Okay. So we begin step one here. You have your actin molecules or proteins hanging out. And next to it, you have your tropomyosin there hanging out as well. Just to make it simple, keep one tropomyosin in the picture. And it's here hanging out. And here is bound to ATP. So let's start here. So the first step we're going to have is step one is called activation of myosin. Activation of myosin. In this step, what happens is ATP. So here, when you begin the process, the myosin is bound to ATP, hanging out there. And then at some point, it will move like this. It will move because the ATPase in myosin will convert ATP to ADP plus PI. So that one step of turning ATP into ADP and PI will provide energy that's used to move the head of myosin this way. What's called activating the head or cocking the head. So now it's in this cocked position. The head of myosin that way. Then step 2 here. So once the head is activated like this, it will now grab actin. So now the head that's cocked will bind to an actin molecule in the area. Okay, so here, this is called cross-bridge formation. Cross-bridge formation is once the head is cocked and the ADP and PI is attached to the head, it will grab and bind. So this formation, so this process of myosin grabbing actin, this is called a cross bridge. So a cross bridge forms between actin and myosin, and then we go to step 3. In step 3, the myosin that's there will release the ADP and PI. And when it releases the ADP and PI, it will move inward this way. So imagine that this called is the number 3 actin. So now it's being pulled this way. Okay, tugging it, because at this point the ADP and PI that were attached to the head of myosin, those two things are released. And when they're released, the head will tug this way, called power stroke. So step 3 is called the power stroke, where myosin will drop off the ADP and PI and then move this way to tug it towards the center. So it does this, like pull it in this way from both sides, okay? And then in step 4, so here you are again, all right. And now in step 4, the myosin will bind to a new ATP, bound to it, and then it will release, drop it off, relax, called detachment. See, it's called detachment, where myosin binds to ATP, and because it binds to ATP, it will then release the actin to detach. And then from there, you come back to here. Activate again, bind it, pull it, detach, activate, bind, pull, detach. That's called a cross-bridge cycling. It happens over and over again. And as it happens over and over again, you keep pulling the head in this way. In fact, each time the head recocks, it grabs a new actin and pulls. A new one and pull like this, like that, to shorten the head. Okay, and that is your cross-bridge cycling process of how the muscle goes from being this way to being this way, being contracted, because it's being cycled in this way by your heads of myosin. Okay, we'll pause there.